In this second video on the immune system, I'd like to continue my consideration of the lines of defense making up the immune system. In the first video, we looked at the skin, which is a primary first line of defense, and the various types of blood cells and what they do as the second line of defense. In this one, we're going to look at the third line of defense and exactly what happens in it. The big difference in the third line of defense compared to the other two lines of defense is this one actually requires or involves the recognition of the particular pathogen or virus or whatever it is that's causing the problem. And the recognition is a chemical recognition. These cells, which are involved in the third line of defense, have the ability to chemically respond to specific foreign chemicals. And that's really, really important. And it's also important to look at what these chemicals might be. If we consider any cell, let's think of this as a cell, the cell membrane of these cells has phospholipid bilayer and proteins dispersed in it. And many of these proteins, say these are proteins in the membrane, many of these proteins have got short carbohydrate chains sticking out from them. These things are called glycoproteins. There's also glycolipids. The very existence of these features on cell membranes is genetic. They are what they are because of the genes in the chromosomes of those cells. These are genetic features. Therefore, they allow these definitions, self versus non-self. So a cell that's in your body that is part of yourself will have the appropriate glycolipids and glycoproteins on its surface. A cell which is foreign, not part of your body, not genetically produced by your system, an invader, if you will, is non-self. And it's that difference which provides the recognition in the third line of defense. There's a name applied to these things when we're talking about the immune system. These things are called antigens. Antigens. Okay, so now let's have a look at exactly what goes on with the third line of defense and the recognition. There are three types of lymphocytes. And all three of them are capable of recognizing what's going on when there's an invasion of some foreign pathogen or some foreign antigen. So if it's not common to your body, it's called a foreign antigen. One of them is called an NK cell. Natural killer. These cells travel about your circulatory system and they respond to cells or things in your blood that they say, well, that's not supposed to be here. So they can recognize self versus non-self just on the basis of what the membrane has on it. And these will release cytotoxins to destroy foreign cells with foreign antigens. One example might be if we consider cancerous cells. The natural killer cells play a very, very big role in destroying cancerous cells. It's very possible that each of us has cells which go cancerous and they're traveling around our bloodstream occasionally. Natural killer cells will recognize them because they don't have the correct structure in their membrane. It may be that they actually lack the antigens that are supposed to be there. So their membrane isn't appropriate, it's irregular. And cancer cells in particular, because cancer cells are produced very rapidly. They don't have time to specialize, they don't have time to mature to become functional cells. So they're non-functional cells that are traveling around the body and they're recognized as being foreign. So natural killer cells get rid of cells like that. The other two kinds of lymphocytes have an even more complicated pattern for the immune system. So let me just erase this and we'll deal with them one at a time. There's the T cells. The T stands for thymus. The thymus is a neck gland located in the neck, and it's there at birth and it lasts for several years. By the time a person goes through puberty, their thymus is no longer there. It sort of disappears. It's no longer functional and the tissues are used otherwise. But in the process, it produces these T cells. They originate in bone marrow, like all blood cells, but the cells go into the thymus and come out as T cells. Now there's two kinds of T cells. There's a helper T cell, Sometimes it's abbreviated this way, TH cell. These cells have got receptors on their surfaces, so they're white blood cells, they're lymphocyte white blood cells, they have receptors on their surface, and they interact with another cell. I have to show you what that other cell is. If you remember back to the types of red blood cells, we mentioned a macrophage. Macrophages are cells which are produced by monocytes, or from monocytes. They develop into macrophages. These monocytes are the amoeboid type cells which can travel around the body, and they'll get into the system, out of the blood system and into tissue spaces and develop into macrophages, so they become very large cells. Macrophages have the ability to engulf things. These are all phagocytic. They'll engulf an antigen, a foreign particle rather, a pathogen, and take it apart, just like the other cells do. But in the process, these macrophages will take parts of the pathogen and they mount it on their surface of their membrane. So it has these things. It'll take parts of that membrane, of this thing, put it on its surface. So these things basically are foreign antigens. It came off a pathogen. And these helper T cells have receptors that identify those. They actually bond onto them, say, wait a minute, the macrophage presents, it's called an antigen-presenting cell, it presents the antigen, and the helper T cell says, okay, we've got it here, we recognize it, and in response to that, these helper T cells do a number of things. First of all, they'll divide because it stimulates cell division, it's mitosis, and they'll release cytotoxins, the same as the other cells do, and other types of proteins. So in addition to being stimulated to divide, mass produce themselves, they're releasing cytotoxins, which once they recognize, okay, that's what we're trying to destroy, is cells that have that antigen, these cytotoxins will destroy the cells that have those antigens. They'll also release other immunoglobins, like interferon, interleukin. These other proteins, these other immunoglobins have specific roles as well. Interferons, for example, they'll be released into plasma and they go out to healthy cells and they will sort of mask healthy cells so these invading particles, these foreign antigens, can't get to those cells. Interleukins are other messengers that once they're released, they can communicate with other immune cells so that the whole body becomes recognizes that there's an invasion or an infection of some sort. The other thing that these do is they stimulate B cells. I haven't said anything about B cells in particular. B cells and another type of T cell, a cytotoxic T cell. And of course, cytotoxic T cell is abbreviated that way, same as helper T cells abbreviated that way. Cytotoxic T cells do exactly this. They release cytotoxins and they destroy it as best they can. Cytotoxic T cells. This response, this type of immune response, where the cells are doing the job, the cells themselves, this is called cell-mediated immunity. This whole thing, cell-mediated immunity. That is one of the components of the third line of defense. The last component of this third line of defense has to do with these B cells. Okay, I haven't said much about them yet. We've got the T cells, we've also got B cells. So allow me to erase this. I'm going to replace that with B. They don't come from the thymus, but they do come from something that's called B, or begins with a B. The B cells come straight from the bone. They don't go through a gland like the thymus to mature and become specialized in any way. B cells are not involved with cell-mediated immunity. These are involved with antibody-mediated immunity. So we've got two types of mediated immunity here. With this particular situation, when these are stimulated by the helper T cells, that causes them to divide. So they're mass produced in the body, in the plasma. One of the things that's different about these cells than other cells is their surface membranes have got a different kind of protein on it. Their surface membranes have got Y-shaped proteins on it. These Y-shaped proteins are antibodies. Now, let me show you a little bit more closely what these proteins are like. If we look at a Y that goes underneath, but I can't draw it underneath very well. That's what it is. If you look at it, you see there's actually 4 proteins here. There's 2 long strands, this one, and then there's 2 short strands. On the ends here, these are receptor sites. So there's 2 receptor sites on each of these things, which is called an antibody. Now don't get them confused with antigen. Antibodies, antigens are very different. This is an antibody. These receptor sites, just by virtue of the amino acid arrangement here, they have a particular shape to them, and they are able to bond or bind with antigens. And there's literally hundreds and hundreds of different antibodies with different amino acid arrangements here. So what happens quite simply is when they divide, they're stimulated to release antibodies. Now the pattern's the same. All these cells are releasing proteins, whether it's interferon or interleukin or cytotoxin or whatever, they're all proteins. This particular type of protein is an antibody and it's Y-shaped. And what it does quite simply is it'll locate a foreign cell and it'll bind onto it with its receptor site. And each of these can bind to two cells. So what happens if you think about it, if you look at this thing on a larger scale, if you get antibodies and lots of these foreign cells bearing the foreign antigens, you'll get these cells clumping together. This clumping together is called agglutination. And what it does quite simply is it gets these foreign antigens, pathogens, or it could be a viral cell, whatever it is, and it clumps them together, and then the other cells, which are phagocytic in nature, they can engulf these things and destroy them. So that's antibody-mediated immunity. There's one more part to this story, and I'll just put it up here. It's the business of memory cells. There are T cells, which are memory cells, and there are B cells, which are memory cells. Those are the cells that are left over afterwards. I mean, in both cases, they're mass-produced. And in both cases, the cells don't all die, or there's leftover cells. The B cells, the leftover B cells remain in the plasma. The leftover T cells, the memory T cells, I should put memory here, memory T cells and memory B cells, these actually go into the lymph nodes. Now maybe things are coming together a little bit in this topic. If you get an infection and your body learns to fight that infection, you survive it, should you encounter that infection again, that pathogen again, your body already has the memory cells. It can already deal with it. It doesn't have to go through this whole recognition process where they are presented with the antigen and so on and so forth. They've already got the B cells, they've already got T cells. The presence of these T cells in the lymph nodes, if you think back to lymph nodes picking up excess fluid from the body and draining it back to the circulatory system, that fluid goes through the lymph nodes. So the memory T cells actually clean, if you will, or take the pathogens, anything harmful, they'll take it out of that plasma-like fluid before returning it to the blood. And I think we better do one more tiny little piece. Memory cells, that's good. But I think maybe we should mention something about vaccinations. If you get a vaccination, you're getting a needle of some sort, more than likely. What is it you're actually getting? What's in that solution they're injecting into your arm? Well, it depends on what you're being vaccinated for. In some cases, a vaccination could contain antibodies. In that case, you're getting what's called passive immunity. The vaccination is giving you the ability to fight whatever disease it is. In other cases, the vaccination is not an antibody, but it's actually what's called an attenuated pathogen. So it could be the bacteria, or it could be the virus, or it could be something, but the word attenuated means that it's been treated. It's been killed perhaps, or it's been taken apart perhaps, or it's been added to some other molecule so that it can't be harmful in its original form. So a vaccination could be an attenuated pathogen. If you get a vaccination with this type of thing, you're actually going to go through this whole process because your body's going to say, wait a minute, that's not supposed to be there. And your body's going to recognize it chemically, and you will generate your own immunity. This is called active immunity. Active immunity is much better than passive immunity because you own it. It's part of you. Passive immunity, it only lasts as long as the antibodies last. Active immunity, it's with you because you've got your own memory cells and all that system is totally set up. That concludes our look at the immune system. We have to take this information and use it one more time though, because in the next video, I'd like to consider blood types and transfusions and why some of those work and some of those don't work. And that topic is very heavily reliant on antibodies and antigens. Thanks for watching.